Long before the Industrial Revolution, our ancestors were already living in environments heavily contaminated with toxic heavy metals. We know this because chemical analysis of fossilized hominid teeth reveals distinct microbands in the enamel and dentine, locking in a record of episodic lead exposure. This exposure isn't a recent anomaly. It stretches across an immense timeline, affecting early Homo species and Neanderthals anywhere from 100,000 to nearly 2 million years ago. The contamination came from natural geological background sources. Hominins consumed water from lead-rich aquifers, and they handled, ground, and applied mineral pigments like ochre for symbolic and practical use. While elevated compared to modern unpolluted baselines, these exposures were largely chronic and sublethal. Populations were not suffering acute fatal poisoning, but they were carrying a persistent toxic burden. If lead exposure was a persistent ecological variable for over 1 million years, we have to ask how that much continuous neurotoxic stress shaped human cognitive evolution. In modern medicine and public health, the consensus is unambiguous. Lead is a potent, harmful neurotoxin with zero safe level of exposure. Elevated blood lead levels in contemporary populations correlate strongly with lowered IQ, impaired executive function, and severe behavioral dysregulation. This presents a biological puzzle. If an environmental factor is purely harmful, natural selection typically penalizes the lineages exposed to it. Yet the hominin lineages living in these contaminated niches survived, expanded, and developed complex cognition. Margolin's paper introduces evolutionary trauma, proposing that long-term neurotoxic exposure functions as a selective filter for an entire species. This framework shifts the investigation from random pathology toward a directional evolutionary pressure that forced early neural systems to adapt. The prefrontal cortex, the region responsible for planning and executive control, is highly metabolic and develops slowly, extending well into adolescence. This prolonged development leaves it uniquely vulnerable to toxic interference. In a healthy neural synapse, calcium ions flow through voltage-gated channels to trigger neurotransmitter release. But lead ions have an identical ionic radius, acting as imposters that hijack these channels. Lead also directly inhibits NMDA receptors, breaking down the synaptic plasticity essential for learning. The disruption extends to white matter. Lead inhibits the maturation of oligodendrocytes, the cells responsible for wrapping axons in myelin. This severely delays the myelination required for fast neural communication. Delayed myelination specifically warps the timing of highly distributed networks, heavily impacting the dopaminergic frontostriatal loops that govern reward, attention, and motivation. Lead does not inflict generic, uniform damage. It systematically targets the exact molecular plasticity mechanisms and timing circuits necessary for advanced human-level cognition. To understand how hominin populations adapted to this specific molecular damage over millennia, we need a mathematical model. This state space, labeled C, represents all possible brain network wirings. A growing brain predictably rolls down this slope into a deep central basin, the ancestral pattern. But a toxic shock field of chronic lead exerts pressure, drastically warping the slopes and shallowing the entire landscape. Because of varying genetic backgrounds, individual brains will take different trajectories through this distorted landscape. Some lack the buffering mechanisms to cope and fail to develop functional networks entirely. By tracking where the surviving trajectories settle in this landscape across generations, we can determine if a species merely survived a toxin or fundamentally evolved because of it. This brings us to two competing hypotheses. The first is scenario A, the null model, defined as survival under damage. In this model, the toxin acts strictly as a culling mechanism. Vulnerable trajectories fail, but survivors are simply lucky enough to build the exact same ancestral brain pattern despite interference. Mathematically, the evolutionary shift in pattern is near zero. The population just endured. A second possibility is scenario B, true pattern phase adaptation. The chronic shock field deforms the landscape so severely that the ancestral basin collapses. To survive constant disruption, the population must discover new computational strategies, settling into a completely new basin, representing a distinct brain architecture. We see this dynamic elsewhere in evolutionary biology. The sickle cell trait, for example, alters red blood cells in a way that confers partial protection against malaria, but at a severe biological cost. Applied to the brain, the neurotoxin acts like malaria. It exerts a selective pressure that favors an architecture sacrificing raw metabolic efficiency for extreme robustness against ongoing synaptic damage. Under Scenario B, the hominin brain's defining architecture is a byproduct of that toxic exposure. If our brains underwent this pattern phase shift, we should be able to find the fossilized evidence of that adaptation in modern human neural architecture. This network graph compares white matter connectivity of a standard primate corticostriatal loop against a modern human equivalent. The human model shows massive expansion in modularity. Look at the frontoparietal tracts, featuring an extraordinarily dense web of redundant alternative routes between key hubs. When synapses are prone to failure, a system requires redundant pathways to keep a signal moving. This specific topology, multiple alternative routes between key hubs, mathematically increases a network's robustness against localized interference. Our uniquely human capacity for flexible, highly distributed executive function might owe its existence to the sheer necessity of routing around chronic generational neurological damage. The authors explicitly state that this pattern phase hypothesis is a theoretical proposition. It is a structured model designed to be tested, not an established fact. Falsifying it requires specific research programs, running simulated lesion models on connectomes, cross-referencing artifacts with isotopic signatures, and isolating epigenetic markers for detoxification. And there is a critical guardrail here. This framework does not imply that damage equals genius. It does not suggest that lead exposure is in any way beneficial to the individual. Evolutionary adaptation operates over millions of years. It cannot keep pace with the rapid, intense, and overwhelming industrial lead exposures found in modern environments. Lead remains an unambiguous public health hazard, and the imperative to eradicate human exposure is absolute, regardless of what subtle adaptations may have occurred in the distant past. But by integrating dynamical systems math with neurotoxicology, the evolutionary trauma framework gives us a powerful analytical lens. It allows us to investigate how hostile environments, whether toxic, viral, or climatic, ultimately act as the sculptors of